Okay, so I mean, if you guys want to go ahead, I will take care of passing the slides.
Okay.
Okay.
Ok.
Let's do it.
Susana.
Susana, you want to start?
Érica, está tudo certo?
Okay, here we go.
Então, después de la emoción de comenzar la presentación, dear professor, thank you very much to be with us today. It's a pleasure to have you here in the Carlos Chagas Filho Institute of Biophysics from the Federal University of Rio de Janeiro. Yesterday, we commemorated the 100 years from our university, so it's a pleasure to celebrate with you with your presentation. Please feel at home, and Professor Van der Leeuw, please, can you do the introduction for our professor?
Okay. Yes, thank you very much, Adolfo, to accept our invitation. It's really a great in a Congress of Microscopy in Ribeirão Preto, São Paulo State, when he showed the first results where glycoproteins could be detected on the surface of a parasitic protozoa. This was really a very important work, and people working with the other parasites start to do similar analysis. And then the glycocalyx was described for parasitic protozoa. At that time, that was a very important conference where we had all people work with membranes. In addition to Adolfo, we have Pedro Pinto da Silva, that was at that meeting, Don Fawcett from Harvard University, Karl Novick, and David Robertson, that was the author of the classic model for the membrane structure. Immediately after his talk, I asked to go to his lab. And in 1975, I had the opportunity to work for a few months in Adolfo lab in Cinvestave in Mexico. And since then, graduated in medicine in 1964 at the Universidade Autônoma do México, then got his master's degree in the Keene University in Kingston, Canada, and then the doctorate at the Faculty of Medicine of UDA. Then he did a postdoc in France in the Institut de Cancer in Villejuif, where there was a famous group working with microscopy and cancer at that time. In Mexico, he was investigated at the National Institute of Cardiology, and then at the Centro de Investigación de Estudios Avanzados in the Department of Cell Biology, and more recently, the Department of Infectomics. Adolfo Martinez-Palomo is one of the most distinguished Latin American scientists. He was president of the Mexican National Academy of Medicine, president of the Mexican Academy of Sciences, director in Mexico for the program of the study of parasitic diseases, supported by the Rockefeller Foundation, then MacArthur Foundation, and the World Health Organization. Director of the Health Council for Science and Technology of the Mexican Presidents of Republic. Scientific Director of Miguel Aleman Foundation, that, as I mentioned when I introduced Dr. Guadalupe, I remember he showed in his last slides that part of her work was supported by the Miguel Aleman Foundation. He is member of the scientific councils of important institutions such as the health science areas of Harvard University, Rockefeller Foundation, the Mexican President of Republic, in recognition of his work. And it's very important to say that Adolfo is member of the National College of Mexico, Colégio Nacional de México. This is a very important institution. There are only 40 members that belong to this National College, and the previous members include personalities such as Carlos Fuentes, Diego Rivera, and Otávio Paz, that got the Nobel Prize of Literature in 1995, only to mention some of the members of this National College. I had the opportunity to visit this college twice and indeed participate of a symposium organized by Adolfo Martinez-Palomo a few years ago. His work, the main focus of his work, is to understand the biology, the structure, and the interaction of anaerobic protozoa with epithelial cells, mainly Entamoeba histolytica, Giardia intestinalis, and Trichomonas vaginalis. So that's really a great pleasure to have Adolfo here. He published around 340 papers, 6 books with an H-index of 50, and got prizes like the Carlos Slim Foundation prizes in health sciences last year. This is probably the most important prize in Mexico in recognition of the work did by Mexican colleagues in different areas. So that's a great pleasure to have Adolfo here, and we are waiting for your talk. Please, Adolfo.
Thank you very much, Vanderlei. As you have heard, Vanderlei is my friend. So that's why he has exaggerated many things about me, but the thing that is sure is that we have a very close friendship that started many, many years ago, as he has mentioned. For me, it's a privilege to congratulate the Instituto de Biofísica, Carlos Chagas Filho, for this commemoration, the Universidad Federal do Rio de Janeiro, and it's really a privilege to participate in this symposium. Thank you very much, Vanderlei. We can proceed. Vanderlei asked me to talk about our main concern for the last 50 years, amebiasis. And we will go from disease to molecules, and I hope that you will see why I have chosen to start with the disease. Can I have the next slide, please? First, I'm going to refer to the main layout concerning disease. The next. First of all, I make a brief introduction, the life cycle. Next. The clinical manifestations, very short. Again, and the global distribution. Next. And finally, concerning the disease, we will look at the scientific output in terms of amebiasis during the last 50, 60 years. Can I have the next one, please? And also as a health problem. Amebiasis, as you know, is the infection of the human gastrointestinal tract by Entamoeba histolytica, a protozoan parasite that is capable of invading the intestinal mucosa and may spread to other organs, as you can see here in this slide. Mainly the slide we will see Entamoeba histolytica, the trophozoite, in thin section. As you can see, it has very complex cytoplasm. There are no Golgi apparatus or endoplasmic reticulum obvious, no mitochondria, obviously. Can have the next. And when you look at the scanning electron microscope, what is evident is that each amoeba is different to the next one. It's impossible to see two who are exactly similar. In fact, that's why they are called amoeba. Amoeba means amoeba, no form, because they change constantly their form. The way of transmission is not the trophozoite. We are looking at the trophozoite, but the cysts. In the next slide, we can see a section of cyst with a typical 4 nuclei, and the cyst as seen by scanning electron microscopy. Now the trophozoite, next one, invades the colonic mucosa and causes dysentery. And here you see a specimen from a human autopsy of a person that died from amoebic colitis. And as you can see, the destruction is evident, and the bleeding caused by the ulceration caused by the amoebas. But then the amoebas may spread via the bloodstream and may give rise to extra-intestinal lesions. Next, mainly liver abscess. Here you have a liver abscess obviously from a patient that died as a consequence of amebiasis. And well, this liver abscess that leaves very little of the liver in that poor patient that died as a consequence of this enormous amoebic liver abscess. The trophozoites may go to other organs and may invade the brain, the skin, the lung, but it's mostly colitis and the amoebic abscess, the most common clinical presentations. This condition, the liver abscess, which results from the migration of the amoebas from the colon to the liver through the portal circulation, is 10 times more common in adults than in children, and 3 times more frequent in males than in females. Can I have the next one? A problem with amebiasis has been to know exactly how many people die as a consequence of amebiasis. So 2 days ago, I asked one of my friends at the Institute for Health and Metrics and Evaluation at the Washington University in Seattle 30 years ago. This is, I got it only 2 days ago from my friend Rafael Lozano, a Mexican that works at this institute. And as you can see, there are 2 things that are important. First of all, amebiasis at present is concentrated, at least in 2017, was prevalent in African countries. In America, practically it's very little. At present, in Mexico, it was one of the major medical problems in Mexico, but it's no longer, although we do have, as you will see, still many cases of liver abscess. And the interesting thing is that if you go to the figures, in 1990, the estimate was 80,000 people in the world died as a consequence of amebiasis. But this trend is slowly lowering. 2000, it was 75,000. In the year 2010, it was 64,000. And the most recent is about 50,000. 100,000 people die still as a consequence of amebiasis. Mostly people die from amoebic liver abscess. Can I have the next one, please? These figures, one thing that has to be taken into consideration is that even though the number of deaths caused by amebiasis is lowering, it is more significant because we are increasing tremendously in terms of population. One of the major causes of death in developing countries, but still is responsible for, is the fourth leading cause of death due to a protozoan infection after malaria, after Chagas disease and Leishmaniasis, and is the third cause of morbidity, again after malaria and trichomoniasis. So it's still a very important infection. Next, please. We did a survey 2 or 3 years ago asking whether there were recent reports on amoebic liver abscess, and we found something interesting. There are still cases everywhere, mostly in, as we have seen in the map, in India, in Bangladesh. In Bangladesh in particular, and India are the two countries that have reported frequently many cases of amoebic liver abscess. Not necessarily the mortality, this is morbidity. Probably the most striking figure is that in the USA, it's the highest figure. Nearly 900 cases have been reported in 2011. Obviously, most of them are not Americans, are Mexicans or people from Central America that have to migrate to find better opportunities in the States, and of amoebic liver abscess. The mortality is very low if they are treated properly in a hospital. The next one, please. And very recently, there was a paper addressing particularly the funding agencies about not neglecting amebiasis and insisting about the number of people that die and the importance of amebiasis as one of the top 15 causes of diarrhea in the world. I agree completely with our colleague Shirley that produced this only a few months ago at the end of last year. Next one, please. This is interesting. Somehow I got into PubMed for this lecture, and I found that we could compare the scientific output of amebiasis and Entamoeba histolytica in the literature. As you can see, before the Second World War, practically there was no interest whatsoever in amebiasis. Practically nothing was written about amebiasis, very little. But then we have a tremendous output, interest in the upper graph on the disease, and in the lower graph on the protozoan itself. Now, what I would like to comment with you is, I mean, with all this output, with its hundreds and hundreds of papers, have made a difference in terms of addressing the medical importance of amebiasis. Because in spite of its medical importance, amebiasis is not a priority for WHO or for the few funding agencies interested in parasitic diseases. Next one, please. Now, I will spend a few minutes talking about amebiasis in Mexico. The reason is that for you in Brazil, your main infectious disease, obviously Chagas. For us in Mexico, it used to be Amebiasis. And I will tell you very briefly some of the historical details. For example, in 1937, a very distinguished group of physicians decided that they had to tackle the problem of amebiasis because it was very common to find people with amoebic liver abscess. When I did my internship in the 60s, all general hospitals were filled with people with liver abscess. Now, practically, it's difficult in large cities like in Mexico City to find a case of liver abscess in Mexico. But 50 years ago, it was extremely common. Now, when these physicians decided to produce a publication on amebiasis, they asked the famous, next one please, painter Diego Rivera. And Diego Rivera went to one of the general hospitals in Mexico, and he was so impressed, very impressed by the poverty and the poor conditions of the people that he saw in the wards of the gastroenterology in the general hospital, that he drew this sketch, which reveals that amebiasis is clearly a disease of poverty and undernourishment. This was a paper sent to the King Carlos IV, the King of Spain, when Mexico was a part of Spain, talking about the obstructive inflammation of the liver, which most probably was liver abscess. And this is 18th century. The 19th century, again, next. Can I have the next? It's the same. No, the previous one. It's the same. The most important clinician of that time, Dr. Jimenez, was concentrated on the study of liver abscesses. At that time, nobody knew that it was caused by an amoeba, but obviously he was referring to liver abscesses. So starting in the 17th, the 18th, the 19th century, up to the 20th century, amebiasis was one of the major problems in Mexico. Now, this interest in, can I have the next one, please? The interest in amebiasis continued because Mexico was the only country that organized, starting in 1970 up to the present time, a total of 20 people, from molecular biologists to epidemiologists, concentrate every 2 years or every 3 years to talk about the recent developments on amebiasis. And I think that this has been very important in terms of attracting to the international audience of researchers the importance of doing research on amebiasis. The next one, please. Now, I will speak to, you saw the hundreds of papers on the disease, but there are three areas in which there have been major breakthroughs to better treat and better know amebiasis. The first one was the distinction between Entamoeba histolytica, the invasive form of amoeba, and a non-invasive Entamoeba dispar. Then the development of the drug that is sufficient for treating invasive amebiasis, and then the advances in imaging. Can I have the next one, please? The story goes back to 1925. The most famous parasitologist of that time, Emil Brundt, published at the Bulletin of the Academy of Medicine in Paris, that there were differences, very clear differences, from amoebas taken from liver or intestinal lesions in comparison with most amoebas that were found in carriers. That means people that had the intestinal infection with an amoeba, but that this amoeba, he insisted, was introduced to Professor Emil Brundt, and Brundt made some seminal contributions to several parasitic diseases. His laboratory in Paris received many people from many areas of the world. And unfortunately, he died in 1951 from a relatively unknown disease. Can I have the next one, please? He died as a consequence of Rocky Mountain spotted fever, which even here in the slide, according to CDC, is one of the deadliest tick-borne diseases in the Americas. So it was very sad that he contaminated himself in the laboratory and he died as a consequence of this rickettsiosis. Now the thing, can I have the next one, that is interesting is that obviously Brundt published in France, and my impression is that people working in the Anglo-Saxon world didn't know French or were not interested in French papers. The thing is that many years elapsed until what we have here from this book, the concept of two different species of amoebas was forgotten until the 70s, when research in Mexico explored this difference. This difference was found by our group, next, in this paper, 1973, and we found, comparing pathogenic strains from what then we knew, later we knew it wasn't Amoeba dispar, that one could distinguish them by the agglutination by electing concanavalin A. This was a very simple observation, but nevertheless it was published in Nature, so I thought, wow, it's very easy to publish in Nature. Well, we were wrong. This wasn't the only the only case. The thing is that almost 50 years have elapsed, and nobody paid attention to the fact that there were two types of amoebas. Now, there were laboratories from several countries, from very different areas, doing enzyme electrophoresis, doing immunoassays, and finally, next one, we came to an agreement. But nevertheless, up to 1997, there was much discussion on all these countries in 1997, and we produced, next one please, this report, which has been taken somehow as the confirmation that Entamoeba dispar is really a separate species of amoebas. As I mentioned, this was the result of many people from many countries coming here to Mexico and agree that there were, evidently there were two different species of amoeba. Right now it seems obvious, but it took a long time to convince people, and this changed radically the epidemiology of the disease. The next one, please. Now, let me introduce you, for those that have not seen them. Here is the culture. Obviously, a major breakthrough was the cultivation under axenic condition by Lou Diamond at the NIH of not only of Entamoeba histolytica, but also of Entamoeba dispar. And this has allowed the comparison of an invasive species. Many more cytoplasmic vacuoles, and there are other characteristics, but they are not sufficiently clear in order to allow for a diagnosis simply by looking at the microscope, even in preparations that have been stained with various stains. If we look at the next one, please. At the scanning electron microscope, they tend to be very different. Entamoeba histolytica is somewhat larger, tends to be more rounded, whereas Entamoeba, the next Entamoeba dispar, looks more like a banana. But again, the difference, the morphological difference, even at the scanning electron microscope, are not so clear as to allow with certainty to distinguish one species from another, which can be done obviously now by a number of immunological and molecular biology means. Now, next one, please. Now, the next breakthrough that made a difference, a huge difference in terms of treating invasive amebiasis, was discovered, and there is very little talk about this. In 1950, the French laboratories, Ron Poulenc, decided to go. They didn't care about the amoebas, and they discovered the metronidazole, and this is the brand name is, as you know very well, Flagyl. And this has been on the market for trichomonas for 70 years. But something interesting happened. The researchers working at Ron Poulenc didn't have at hand a good experimental system of trichomoniasis. And instead, can I have the next one? They had an experimental model of amebiasis, of intestinal amebiasis. So they decided to check for the effectivity of metronidazole first in the amoebas, and to their surprise, it was as effective, or probably more effective, for amoebas than for trichomonas. So Flagyl was started using for treating amebiasis, and so far it has been the best, probably the only real efficient drug to control invasive amebiasis. And so far, no resistance to the drug has been recorded. Only in the laboratory, using cultures, people have been able to induce a resistance to the drug. But in the clinical setting, there is no resistance to the Flagyl. So Flagyl, the metronidazole, is still the standard of gold, the gold standard for treating invasive amebiasis. Can I have the next one, please? Now, other breakthroughs, obviously, in terms of for people trying to diagnose and to study the patients at the hospitals, was the development of the application of different imaging systems. The ultrasound was the first, and is still the first. Is very useful. It's the least expensive, and it's been used in settings, in pool settings. It's very, very useful to follow the course of invasive amebiasis, obviously mostly the liver, and the holes that you see in the image are obviously produced, in this case, by two different amoebic liver abscesses. But then other more modern have been used, more modern imaging systems like computed tomography, and you see here what a physician would say it's a beautiful liver abscess. It's very well. Be more refined, you can go to NMR. Next one, please. And you see here with NMR one plane, one section of another large liver abscess. Now let's go from the experimental work, experimental pathology. And many laboratories have worked with epithelial cells and with intestinal models and with liver models. And I will show you just a few slides of what we have been doing during the last many years, starting with the interaction between Entamoeba histolytica and culture cells. Can I have the next one, please? Here you see an amoeba on top touching an NDCK cell, an epithelial cell. You see a lobopodia, a site where actin is concentrated. And very rapidly, the amoebas Entamoeba histolytica can produce a rapid death of these epithelial cells, and the whole culture disappears in terms of probably half an hour or a little bit more than that. Whereas Entamoeba, this part produces some damage, but it takes much more, a longer time, and the damage is not as profound as the one produced by Entamoeba histolytica. In the next one, we see how an amoeba, next, has killed an epithelial cell and is trying to engulf this killed amoeba, and the amoeba appears dense to the electrons due to the penetration of an electron-dense stain. But it's obvious the intimate contact of the amoeba and how the amoeba is trying to grab its prey. The next one, in scanning microscopy, shows you how the amoeba tries to suck, if I use this term, the surface of the epithelial cell, and has destroyed part of this monolayer. And this was described by us many years ago, and one of our American colleagues decided that it was his observation until this process, trophocytosis or something like that, and he got published in Nature, but this was published by us about 20 years before. But well, this is how it happens. If you have friends in connections, then you get access to better journals. Can I have the next one, please? This is an old scanning electron microscope, but it's one of my favorites because it shows one amoeba, one single trophozoite, engulfing by phagocytosis 1, 2, 3, 4, 5, 6, 7, 8, and probably more. And it's incredible. And that's how the amoebas become larger when they are in a lesion, in an intestinal or in a liver lesion, because they phagocytize so many dead cells that they increase in size. And the classical parasitologist thought there was a different species of amoeba called magna, because they were huge. But they were huge because they had a tremendous lunge. It's really very impressive when you look at the microscope on the living conditions in vitro, how the amoeba kills and incorporates the dead cells into their cytoplasm. Okay, now this is some of the things that can be done in the laboratory with culture cells. Can I have the next one, please? The next. In the guinea pig, you can see the early stages of penetration of one single trophozoite, disrupting the epithelium, the surface of the mucosa of the large intestine of the guinea pig, and you can see a lobopodia obviously filled with actin, trying to get inside the lamina propria of the epithelium. This is why one of the aggressive factors, one of the pathogenic factors of the amoeba is undoubtedly the locomotion. I don't think that you have seen amoebas coming directly from a liver abscess, or probably it's better from an intestinal alteration in a patient. It's unbelievable, the movement of the amoebas. They literally jump out of the microscopic field. They move extremely rapidly. When you see the amoebas in axenic cultures in the laboratory, they are, dormant. They barely move. You have to use microcinematography to study the movement. But in amoebas taken from real specimens, electron microscopy at some of these experimentally induced lesions in the intestine. Can I have the next one, please? This is what you find. You find first on top the presence, the addition of the amoebas to the intestinal surface, and then the penetration at left in a small ulcer, and then a larger ulcer, and how amoebas are being concentrated on the site of the ulceration. Well, this is so far concerning the intestinal model. What about the liver model? The liver model is probably more interesting. The next one, please. Because the classic text used to say that there was no inflammation in amebiasis. Well, there was no inflammation because pathologists were looking at the end stages of the infection. Whereas here we have found with various of our colleagues, at 6 hours, the tremendous inflammation, as you can see at left, there's a single trophozoite surrounded by hundreds and hundreds of polymorphonuclear leukocytes. And according to our observations, is the death of these leukocytes that initially produces damage to the liver. It's not a direct damage of the amoeba. Damage of the liver is indirectly through the killing by the amoeba of the tremendous output of trophozoites that come in a few hours after the amoebas are inoculated into the liver. So far, so good for the experimental modern models. Now we will go to the last part of the talk, which is the more scientific part in terms of studying the molecules and the processes like addition and locomotion. And I will very briefly mention just an example of each one. Each of these pathogenic factors represent the main interest of several laboratories. There are labs working on lectins, other on pores, other on proteases, other on addition. It's the output. It's difficult to follow the literature because there is, as you have seen, when I show you the graph on the scientific output, it's really important. Lectin addition of the parasite occurs mainly through a surface Gal-Gal-Nac lectin. This lectin binds to exposed terminal Gal-Gal-Nac residues of target cells glycoproteins. And this was described many years ago by Petri and colleagues. And for me, it's surprising because now they have decided to use this lectin that has attracted their attention as a possible candidate for a vaccine against amebiasis. At the end of the talk, in a few minutes, we will talk about whether I personally think that a vaccine is necessary or not. Can I have the next one, please? Now, the next pathogenic factor, which has produced fantastic work from several laboratories, starting with Leipe in Germany, is once Entamoeba histolytica establishes contact with mammalian cells in vitro, a rapid cytolytic event takes place that results in swelling, surface blebbing, and lysis of target cells. And this includes many cells that can be in contact with the amoebas. And this process at the beginning was thought to be similar to the perforin-mediated lysis by cytotoxic T lymphocytes. And then it has been proven that Entamoeba histolytica produces amebopores, and that these amebopores, at least in vitro, is partly responsible for the killing of the cells in vitro. There is some really fantastic work in the molecular biology of the amebopores, and this still continues, even though it was started about 15 years ago. Now let's go to the next pathogenic factor, other factors. Cysteine proteases are by far the most abundant proteases in the amoeba. The amoeba is loaded with proteases. They are encoded by approximately 50 genes and are responsible, and again in vitro or in some models, but mostly in vitro, are responsible, these proteases, for the penetration of the epithelial barrier and mostly for degradation of host extracellular matrix components. And here you have one example from one of the labs at Simvestav from our colleague Mireia de la Garza. The next is we're not talking, the next one is not a molecule. Can I have the next slide, please? Is addition. Addition has been studied in our laboratory by Daniel Talamas. Here you see what Daniel made when he compared addition of Entamoeba histolytica and Entamoeba dispar to fibronectin. The addition process is very complex, and there are several laboratories trying to understand the molecular interactions, the molecular control. Next. And here you see what he has found by comparing scanning electron microscopy with confocal transmission and also atomic force microscopy. Probably the most interesting thing about this slide is to compare in the confocal. What you see in green is the substrate. Where the amoeba interacts, and you see the hole that you see with Entamoeba histolytica is the disruption that the amoeba has produced on the substrate. And well, there are many studies concerning how this is regulated and what is the difference between Entamoeba histolytica and Entamoeba this part. There are some clues, but so far it's not possible to say with certainty what are the basic differences that respond for the fact that Entamoeba histolytica is invasive, is pathogenic, whereas Entamoeba dispar is not invasive. Now let's see to my last comment. Can I have the next slide, please? Our colleague Shirley says, and she's right, amebiasis is a global concern. New innovations for diagnosis, discovery of drug targets, and so far there are no resistant forms of amoebas in the clinical setting. But obviously, it would be good to have another drug as efficacious as metronidazole, and probably with less side problems. But what I don't agree is that we need a vaccine. We don't need a vaccine. I don't think that really the public health experts think that the basis for the control of this infection would be a vaccine. And one reason for this, can I have the next one, is that amebiasis is a disease of poverty. It's a disease related to lack of education, to lack of clean water, to lack of hygienic conditions. And I think that it's not very appropriate to spend millions of dollars for a vaccine where it would be spent much better in trying to reduce inequality and poverty in those developing countries that I show you on the map that are the ones that are suffering and are having thousands of deaths due to Entamoeba histolytica. Here is the typical Mexican sender, the producer of tacos, of our classical meat, classical dish. And obviously he knows that flies, that the food has to be covered from the flies, and he has something in his hand related to poverty. And that's my end message, and I will end. Can I have the next one, please? By thanking, the people that did the work are here, and I'm at the back because I did very little or nothing. But these guys are responsible. Probably, if you see closely, there is a unbalance in terms of gender, because we have many women and a few colleagues, but I think that's very good. And I will end with the next slide, because I want to thank next. These are the people that are really working 24/7, because the projects, these are the ones that spend the best days or one of their best years of their life working on the amoeba or other infections or other diseases that are of interest to our laboratory. And I thank them very, very much. I cannot give their names one by one. Next one, please. So I will end by saying muito obrigado, muchas gracias.
Muchas gracias, profesor Adolfo. Gracias por la interesante charla que usted dio para nosotros hoy. Thank you very much for your talk. And now we have a few questions, if you don't mind.
No, go ahead.
Professor Vanderlei de Souza asked, what determines the pathogenesis of a given Entamoeba histolytica strain? any specific molecule? Is possible to distinguish histolytica, the dish part, using biochemical or molecular markers?
Yes, it is possible to differentiate between the two species of amoebas, and there are kits available in the market for the clinical setting, and they are very useful. They are not very cheap, so it's difficult in some settings, in very poor settings to use them, but they are available. And on the other hand, the pathogenicity of Amebiasis, as I try to explain, is a very complex, very complex phenomenon where you have proteases, where you have reports, where you have the mobility, the capacity to phagocytize of the amoeba and to displace between the tissues. So it's a very complex phenomenon. It cannot be ascribed to a single molecule or a single activity of the amoeba.
Got it. What is presently known about the process of capping of surface ligands in histolytica? It's involved in the process of interaction with host epithelial cells?
Well, I didn't have the time to mention, although it would be interesting because one antibodies or lectins in contact with the surface of Amebiasis histolytica, very rapidly the complex will move towards the posterior end of the amoeba called uroid, and the amoeba will dislodge this antigen-antibody complex, and probably with this phenomenon, the amoeba gets rid of the consequences of the antibody interacting with the antigen. The same happens with lymphocytes, but what is the significance of this in terms of the pathogenesis, we don't know. People have seen capping of amoebas in liver or in intestinal lesions, but we don't know whether it is really a process by which amoeba gets rid of the immunological response, the molecular response of the host.
Professor Maria Cristina Machado Mota asked, amoebas are extraordinarily diverse, significant pathogens, and occupies an evolutionary important position in the tree of life. However, they remain vastly understood. Why?
Yes, well, of course. It's one of the things that fascinated us is that amoeba looks so simple, and in fact, it's so complex. Even if one is not interested in amebiasis, the amoebas represent apparently a very simple cell, but the complexity of the activities of the amoeba, the movement, the different phagocytic and pinocytic capacities, the cell and nuclear division are extremely complex. And our colleague is right. We know very little about it, and that is why amoeba is of interest to many laboratories that are really not interested in amebiasis as a disease, but interested in the amoeba as a useful, apparently simple model to study some of the basic cellular functions, such as division and movement and addition.
The fact I work with phagocytic cells in the host, and we have extraordinary data when you compare phagocytosis and the mechanism of amoeba with phagocyte cells. So I think it's interesting work, and it's a pity that not so many people work with amoeba.
Yeah, amoeba is acanthamoeba are also very, very interesting. We have also done some experiments. When I mention we, our colleagues have done it at the laboratory. But I think that Acanthamoeba is also fascinating, as interesting as Entamoeba.
Do you know if we have data published with Entamoeba an interaction with another bacterial or fungi cells in the gastrointestinal tract?
That's a very interesting question because we don't know, at least I don't know, what is the extent, the importance of the bacteria in the gut in terms of exacerbating explore. I mean, when we say nobody has explored further, is the fact that amoebas have viruses in the cytoplasm that look like rhabdovirus. What are these viruses doing in the cytoplasm of the amoeba? Even in amoebas found in human lesions, we don't know. But there is no doubt that amoebas may harbor not only bacteria but also viruses.
Professor Van der Leek asked, are you studying Naegleria, especially the process of transformation for amoebic into flagellated cells?
No, I didn't understand the question. What was?
If you study Naegleria, especially the transformation from amoebic to flagellate cells. Si você estudia Naegleria y sobre todo el proceso de transformación del estado ameboide para el estado flagelado.
Oh, that's even more complex than, no, we haven't done it. But obviously this is even more complex problem if it's difficult to study the amoeba by itself and the encystation process in what you're mentioning, transforming from one morphological form to another completely different, is very complex, and we have not done anything in that respect.
There are many data about the virulence factors that modulate the severity of amebiasis, but the intestinal microbiota can also contribute to the severity of the disease. It can promote the secretion of thin mucus protecting the mucosa.
Probably. I think that only recently the importance of the mucous layer has been into the attention. The thing is that we don't really have good intestinal models, and people are trying to devise better models to study the intestine, but there have been some advances, but still we are far ahead to understand the very complex interaction from the time that an amoeba multiplies in the intestinal, in the colonic lumen, and gets in contact with the epithelium, first with the mucosa and the epithelium. It's a very complex process, and obviously lectins have much to do with this first step.
Professor Cristina Mota asked, and about the presence of symbionts in amoebas, do they increase the parasite's infectivity? The fact that some amoeba can be with symbionts.
Yes, but I mean, this is very interesting, but so far I don't know, that's my ignorance, but I don't know of any studies in that respect, at least recent studies. But this is something that may be very interesting to think how the environment may enhance or inhibit the virulence of the amoeba.
We have another question from our students, and they ask the fact that the cyst can survive in the intestine of some arthropods will have an epidemiological importance. Epidemiological.
No, I don't think so. So far as we know, and well, these decades have been studied the epidemiology of amebiasis. The human is the only species responsible for the transmission of the disease. One thing that I didn't mention, and it's interesting, why have you many chagas in Brazil and you don't have amebiasis or very little? This is a question because, as you have seen, amebiasis is very common in many African countries. So what's different in the Brazilian environment, that I'm sure that you have Entamoeba dispar, and you have some Entamoeba histolytica, but as far as I know, there is very little the invasive amebiasis is not important as a major public health problem in Brazil.
Do you think that cryptosporidiosis and amebiasis should be included in the list of neglected diseases?
I'm not sure. I think that WHO is right because, I mean, obviously, all of us working on amebiasis would like to have money for our research from the funding agencies. But I think that WHO is right because the amebiasis is a disease of Chagas and other parasitic infections. Obviously, you have drugs for, but not as effective for what, 50, almost 60, 70 years. Metronidazole has proven effective. So if you have that, and you're interested in reducing morbidity and mortality of amebiasis, you have to go to the social consequences of disease, not the biological, or not modifying other alternatives like a vaccine.
So thank you very much, Professor Adolfo, for your excellent talk in the name of the Biophysical Institute. Muchísimas gracias por la charla que el señor dio para nosotros, poder aprender un poquito más sobre amebas. Pido disculpas de nuevo en el nombre del profesor Vanderlei, de los organizadores del simposio, por los problemitas técnicos que tuvimos en el principio, pero al final todo dio cierto y el señor consiguió dar esa tremenda y fabulosa charla para nuestro instituto y para la conmemoración de los 100 años de la Universidad Federal de Río de Janeiro. Muchísimas gracias por su atención, por su cariño y por la paciencia.
Muchas gracias. Vanderlei, ¿me escuchas?
Sí, claro. Agradeço e lamento não poder ter ido a México. Estava planeando ir este ano, pero com a pandemia foi interrompida todas as viagens, pero espero que no início do próximo ano eu pueda ir, como também Daniel pueda volver ao Brasil para completar alguns estudos que iniciamos. Por favor, mis saludos para os amigos como Viviana, Arturo, Daniel, Vitor, e sempre me recuerdo los anos de 75, 76, quando fui a México várias vezes, e grandes amigos que já não teremos mais, como Carlos Arguello, com quem iniciamos estudos da carga da superfície del tripanosoma, seguindo a linha que havias hecho com Entamoeba histolítica. De modo que te agradeço, Adolfo, por la conferência e os famosos encontros de amebiasis que hablaste. Continua havendo todos os anos, porque eles se pensaram quase que simultaneamente com os que temos em Brasil, que são os encontros para a enfermidade de Chagas, que depois se ampliou para a leishmania, e que hoje é a Sociedade Brasileira de Protozoologia, que faz seus encontros todos os anos. Este ano está um pouco mais complicado, pero tradicionalmente mantenemos los congresos de protozoología.
No, sí, yo creo que esto, lo de los seminarios de amebiasis siguen. y seguramente el año que entra habrá uno. O sea, nosotros ya no estamos en el laboratorio responsabilizados de los seminarios, pero sí siguen. Oye, Vanderly, y los que te esperan en México son mis hijos, para que juegues fútbol con ellos, como jugabas fútbol a los años 70.
Sí.
Ahora querrá ser solo portero. Ellos todavía juegan. ¿Te acuerdas que jugaste con ellos?
Sí, me recuerdo, pero hoy yo ya no juego. Yo espero que la pelota caiga en mis manos.
Bueno, muy bien. Vanderly, te agradezco mucho la oportunidad de estar en contacto con ustedes. Un abrazo para todos. Cuídense mucho.
Ok, gracias.
Hasta luego.
Hasta luego.
Obrigado.
Isso.
Érica